Artificial hybridization. When we studied pollination, we have studied cross pollination. Cross pollination is a natural type of hybridization that we have mentioned earlier. So today's topic is artificial hybridization. It means man-made hybridization. So this actually comes under the chapter Strategies for Enhancement in Food Production. I think you have already studied how to produce a new hybrid or hybrid crop. For example, hybrid sunflower, hybrid jawar, hybrid maize. So there are many steps in hybridization. What is hybridization? The crossing of two male and female parents differing in one or more characters to get new hybrid is called hybridization. Hybridization is carried out or done by a plant breeder. We call him as genetist. At research stations, the hybridization work is going on. So that includes many steps you have studied already. For example, germplasm collection, selection of parents, then hybridization. So, like that, many steps are there. At the end of hybridization, plant breeder produce new variety of crops. Okay. So that's all, a brief introduction. Now, according to our textbook, we shall study the artificial hybridization. The artificial hybridization, as I mentioned earlier, is carried out by plant breeder. Plant breeder means genetist who breed new variety of crops. We call him as genetist also. So the genetist producing new varieties of crops with desirable characters. Cross two different species or genera. Say for example, if two different species are crossed, we call interspecific hybridization. The cross made between the same species, intraspecific hybridization. If the cross made between two different genera, intergeneric hybridization. Like that, there are different types of hybridization also there. Now we shall see artificial hybridization. What is artificial hybridization? The process of transfer of desired pollen on the stigma by preventing contamination of unwanted pollen is called artificial hybridization. See, what is the meaning of this definition? Artificial hybridization is a process in which what happens? Pollen grains of desired plant are transferred. That is, from the male plant, pollen collected and transferred artificially. Where? On the stigma of female flower. This we call artificial hybridization. How means the stigma is prevented by contamination. Contamination means if any unwanted pollen should not fall on the stigma of female flower. Okay. So that's all. Artificial hybridization. The meaning. Then, how to conduct this artificial hybridization? As I told you, many steps are there, but here we are going to discuss only 2 steps. Artificial hybridization is carried out in 2 steps. The first step is called emasculation, and the second step is called bagging. Emasculation and bagging are 2 important steps. Okay. Next, we shall see emasculation. What is emasculation? Or how to define emasculation? Emasculation is a process of removal of stamens or anther from bisexual flower bud or floral bud before dehiscence or anthesis is called emasculation. What is the meaning of this? Emasculation means it is a process. One process in which what we are going to do? We remove the stamens or we remove the anthers in a bisexual flower. You know, bisexual flower consists of stamens and pistil. In emasculation, the stamens are removed. When the stamens are removed, before the anthesis, anthesis means dehiscence of the anther. Dehiscence means opening of the anther. You have studied case of anther. Mature anther, when reaches maturity, the anther split or dehisced pollen grains are liberated. So before the anthesis, we remove the stamens. Okay. How the stamens are removed? There are different methods also there to remove the stamens during emasculation, and depends on the type of flower also. or the crop. Say for example, one step is we call forceps method or scissor method. See, when the flowers are very big, flower bud opened one day earlier than artificial pollination. With the help of a forceps or with the help of a scissor, the anthers or the stamens are removed. So this method is called forceps method or scissor method. So this is used where the flowers are big. Suppose you imagine we are conducting a hybridization experiment in grasses. Grasses means bajra, jawar, ragi, like that. Flowers are so small, very difficult or tedious job to open the flower. So what we do is the whole inflorescence is dipped in water or alcohol. See what happened? The stamens will become sterilized, we can say. This process is emasculation. Say, the emasculation word is equivalent to castration in animals. ಅನಿಮಲ್ಸ್ನಲ್ಲಿ ಬುಲ್ಸ್ನ ಕ್ಯಾಸ್ಟ್ರೇಷನ್ ಮಾಡಿಸ್ತೀವಿ ನಾವು. Emasculation is equivalent to castration of animals. See, during emasculation in a bisexual flower, the stamens are removed. Now, after emasculation, the flower becomes unisexual female flower because the flower consists of only the pistil. Stamens are not there. Next, the female flower is covered with a bag. That what we call it as bagging. It is done to prevent contamination. So that's all about emasculation. In brief, the removal of stamens is emasculation. Next, you shall see the second step, bagging. Okay. Bagging. What is bagging? The process of covering female flower or emasculated female flower with a bag. Bag may be made up of butter paper or polythene bag or even cloth bag is called bagging. Once again, the process of covering a female flower with a butter paper bag, polythene bag or cloth bag is called bagging. What is the importance of bagging? Bagging is done just to prevent contamination. Contamination means the pollen of other plant may deposit on the stigma. To avoid that, bagging is done. In artificial pollination, pollen is collected when early in the morning in a muslin cloth buttonally. Then the bag of female flower is opened early in the morning before 10 or 11 o'clock by a skilled labor. Then the pollen is dusted on the stigma of emasculated flower. This is actually artificial hybridization. What is that? The dusting of pollen on the stigmatic surface. Okay. Thus, artificial pollination is carried out during hybridization experiments by scientists. Okay. After artificial pollination, the female flower is again covered with a bag. It is called rebagging. Rebagging is done. Okay. Then a label is put. Label showing the plant name, breeder's name, date of emasculation, date of hybridization, and approximate date of maturity of the crop. Okay. The pollinated flowers allowed to develop fruits. ಉದಾಹರಣೆಗೆ ಜೋಳದ ತೆನೆ ಮಾಡ್ತಾ ಇದ್ರೆ ಜೋಳ ಕಾಳಾಗೋದಕ್ಕೆ ಬಿಡ್ತೀವಿ 3 months. Okay. You imagine if the flowers are unisexual or the parent is unisexual. Unisexual plant produce unisexual female flower, unisexual male flower. Okay. Is it necessary the emasculation process? No. If the flowers are unisexual, emasculation is not necessary. Okay. Very important need point. If after the emasculation, here also female flowers are bagged. Then with the desirable pollen, pollinate artificially, then rebagged. That's all about bagging. What is the significance of this artificial hybridization? Artificial hybridization is carried out to produce superior quality hybrids. Remember, superior quality hybrids means ಸನ್ಫ್ಲವರ್ನಲ್ಲಿ ಹೆಚ್ಚು ಇಳುವರಿ ಬರ್ತಕ್ಕಂಥದ್ದು, ಜವಾರ್ ಹೆಚ್ಚು ಇಳುವರಿ ಬರ್ತಕ್ಕಂಥದ್ದು, ಮೈಸ್ ಹೆಚ್ಚು ಇಳುವರಿ ಬರ್ತಕ್ಕಂಥದ್ದು. ವಾಟ್ ವಿ ಕಾಲ್ superior quality hybrids. In these hybrids, the yield is high, disease resistant, drought resistant characters are introduced. So that's all about bagging and artificial hybridization. Important for 3 marks. Okay. Explain artificial hybridization. Next is what happens after artificial hybridization. That is, pollination takes place. After pollination, what happens in angiosperms? Fertilization takes place. Okay. Now, we shall see some points about fertilization. After pollination, the pollen is nourished on the stigma. It means the pollen is deposited on the stigma. Stigma secretes some chemical or the sugar or the carbohydrate, and thus it is nourished on the stigma. You know, during two-cell stage, the pollen is pollinated. The pollen is at two-cell stage, now begin to germinate on the stigma. Now the intine of the pollen, the pollen consists of two wall layers, exine and intine. On the exine, small pores or germ pores are present, and exine is thick, intine is thin, made up of cellulose and pectin. Now, the two-celled pollen germinate and produce pollen tube. That is, the intine comes through the germ pore in the form of a germ tube. Now, or meanwhile, the generative cell divide, or the nucleus of the generative cell divide mitotically and produce two male gametes, or the sperms, which are non-motile. Okay. Now the pollen tube goes through the stigma and style enters into the embryo sac through micropyle, then through synergids. Okay. ಪಾಲಿನೇಷನ್ ಆದಮೇಲೆ ಪೋಲನ್ ಟ್ಯೂಬು ಬರುತ್ತೆ. ಪೋಲನ್ ಟ್ಯೂಬ್ನಲ್ಲಿ ಎರಡು ಮೇಲ್ ಗ್ಯಾಮಿಟ್ಸು, ಒಂದು ವೆಜಿಟೇಟಿವ್ ನ್ಯೂಕ್ಲಿಯಸ್ಸು ಇರುತ್ತೆ. ನಾವು depending on the entry of pollen tube into the embryo sac, the fertilization is classified into three types. Of course, this is not mentioned in your textbook. Porogamy. What is porogamy? Poro means opening. So, if the pollen tube enters through the micropyle, fertilization takes place, then it is called porogamy. That is, entry of pollen tube into the embryo sac through the micropyle is called porogamy. It is common type. In some plants, the opposite end chalaza is there. Through the chalaza, the pollen tube enters into the embryo sac. We call chalazogamy. And one more type is also there. In few plants, the pollen tube enters into the embryo sac or the ovule through the integuments. The entry of pollen tube through the integuments is called mesogamy. Thus, there are three types of fertilization are identified: porogamy, chalazogamy, and mesogamy. Okay, now we shall see double fertilization. Very important for 5 marks. Okay, double fertilization. Refer the diagram. See the ovule embryo sac, egg apparatus with ovum, at the center definitive nucleus diploid one, and three antipodals at the callosal end, embryo sac, central cell, the pollen on the stigma, germinating pollen, pollen tube, then the tip of the pollen tube containing two male gametes. You can see in the diagram. Double fertilization. In angiosperms, pollen tube after entering into embryo sac through the micropyle or the chalaza or the integuments, okay, takes place. Normally, the pollen tube enters into embryo sac through micropyle, then enters through the synergids. Synergids contain filiform apparatus that guide the pollen tube to enter. Now, the tip of the pollen tube burst. Burst means open. Cytoplasm is released along with the two male gametes into the synergids. There is a diagram in previous class, the releasing male gametes. You may refer. Okay. Among two male gametes, one of the male gamete fuses with the egg or ovum. This process is called syngamy. Syn means fuse. Gamy means gametes. The gametes are fused. This process is also called fertilization and produce diploid zygote. This we call fertilization. The union of male and female gamete is called fertilization. Okay. After fertilization, the diploid zygote is formed. Okay. Now, in angiosperms, there is another second male gamete is there. The second male gamete moves towards the polar nuclei or towards the diploid secondary nucleus or the definitive nucleus, fuses and form pen, primary endosperm nucleus. This process involves fusion of three haploid nuclei. Hence it is called triple fusion. So fusion of male gametes takes place again, second time. Okay. Triple fusion. See the diagram showing double fertilization. Examination alli baribeku. Now, last point. In angiosperms, two types of fusion noticed. That is the syngamy and triple fusion. So syngamy and triple fusion takes place in the embryo sac. Hence the process is called double fertilization. Remember, double fertilization is a special or unique character of angiosperms only. Neat point. In no other plants or groups, bryophyte, pteridophyte, gymnosperms, gymnosperms, double fertilization is absent. It is found only in angiosperms. And it was discovered by a Russian scientist, Navaschin. Navaschin. Okay. So that's all double fertilization. What happens after double fertilization? The central cell, big cell, transformed into primary endosperm nucleus, which is triploid. The later it develops into triploid endosperm. Okay? The endosperm is formed from the pen. Then the zygote develops into an embryo. So that's all about double fertilization. Very important for main examination and CET NEET. Okay, very important. Thank you.